Okay, good morning, everybody. And today we'll carry on with our discussion about the immune system and try and put it all together today into a clear, coherent story about the adaptive immune response. And before we do that, let's just review for ourselves the different players that we're going to talk about. The first really important player is the dendritic cell. Dendritic cell is a cell which is actually part of the innate immune system. It's a cell rather similar to macrophages. They wander through the tissues, through the body fluids, all over the body. And when they encounter something like a pathogen or whatever, they ingest it, digest it, and display portions of the antigen on their cell membranes. They display it with a special molecule called MHC class 2. All cells have MHC class 1, but only a certain restricted set of cells possess MHC class 2. And they possess the MHC class 2 specifically so that they can interact with other lymphocytes or with lymphocytes in general. So that's our first player, is the dendritic cell. As soon as it ingests something and displays the antigen, it becomes activated and it moves to the lymph node and moves through the lymphatic vessels, actively migrates to the lymph node, and it does so that because it's in the lymph node that it can encounter all of the lymphocytes that it needs to interact with. We are going to talk about 3 particular lymphocytes. There are others, but we are going to just talk about 3. And these are, first of all, the B cells. The B lymphocytes are those which are going to end up producing antibody. The other lymphocytes that we are going to consider are the T lymphocytes, and there are 2 kinds. These are the helper T cells, often just abbreviated TH cells. And their most important interaction is with B cells. They are the ones they kick the B cells into full activation. B cells can be activated on their own, can be activated by antigen on their own, but it's a rather weak, poor response. However, the response to interaction with T cells is very profound, very, very robust reaction once T cells interact with them for reasons that I'll show you in a minute. So the helper T cells are going to interact with B cells to get them to produce good antibody. But there's another really important group of T cells, and these are the cytotoxic T cells, TC cells. And these cells, actually, once they're activated, they can go out and they can move around the body, and they will actively seek out cells which are infected with the pathogen that the B and T cells are responding to. And they will also act against things like tumorous cells, potentially tumorous cells, and they kill them. That's why they're called cytotoxic. They kill in a particular way that I'll tell you about. But they actively kill cells which are infected or which have transformed, which no longer look like self. So those are going to be our main players. Let's have a look.
Oh yeah, so I have a question.
Go ahead.
So wait, can you go back to the other side for a sec that we were just on?
Yep.
So the cytotoxic, how do you say that? I'm sorry.
Cytotoxic.
Okay, so those cells go and kill like tumorous cells that are like infected and stuff.
Correct.
So if that's the case, wouldn't there be like, I don't even want to say a cure, but like, wouldn't there be like a better way for us to cure cancer if we have these cells in our body? Why can't they use those kind of cells in like medicines and stuff?
They can. And they're not the only cells which kill tumor cells. If you may in fact, hear now about immunological treatments for cancer, this is exactly what they're doing. They are culturing these cells. What they do is they'll culture cells like the cytotoxic T cells. They'll culture them and introduce them to the cancer, to features of the cancerous cells, and then put them back into the body. And in some cases, they have had very, very, very spectacular results from doing this. The most famous case, in actual fact, at the moment, is President Carter, and who's, I mean, a very elderly person who developed brain cancer a couple of years ago, and he was treated using T cells, and was almost completely cured. Right. So it can have very spectacular results. You're quite right.
So then, if it's working for people, why don't they release it more to the public and say like this could be a potential cure for cancer? Or
It is somewhat unpredictable at the moment. It's not absolutely clearly understood how to activate these for a particular cancer. Sometimes they have good results, sometimes they have poor results. The other unfortunate thing is it's very, very complicated and very expensive. So it's still experimental, put it that way. It's still an experimental phase, but it's certainly becoming more and more standard to try to develop immunological treatments for different cancers. Also, some cancers respond better than others. That's the other.
okay. Okay, no, because when I was reading the slide and it was saying that it was treating like tumors, like tumorous cells, that
Mm-hmm.
just made me think. I was like, if it's treating tumorous cells, that could be a potential cure to cancer.
Yes, it absolutely is. It absolutely is. It's one of the major lines of research in cancer treatment at the moment.
Okay,
Yeah,
thank you. I just wanted to
sure.
clarify up on that.
Okay, so let's just have a look at how these T cells in particular are activated and how they respond to activation. So here, this here, we can assume it says here antigen present cell. We're just going to presume it's a dendritic cell. It's been wandering in the periphery. It's picked up pathogen, digested the pathogen, and it's just, excuse me, it's displaying portions of that pathogen as antigen. It displays it with this special molecule here called MHC class 2 there, which only a few cells, B cells, dendritic cells, those macrophages, these antigen presenting cells, only a few of them can do this. A helper T cell interacts with the dendritic cell through this combination, the MHC and the antigen. Here's the key. This T cell is one of those lymphocytes that has a receptor that only will react to this particular antigen. The T cell, each T cell is unique in what it will react to. So they are very, very specific, and they're not necessarily that many of them. When the dendritic cell enters the lymph node, it actually sends out messages to all of the T cells in the lymph node, and the T cells actually cluster around and attempt to bind like this. But unless they have the exactly matching receptor, they can't bind. But those that can bind are activated. And what happens is the dendritic cell and the T cell actually talk to one another, but they talk to one another in the form of cytokines, of immune hormones that are emitted. And there's this kind of chatter, hormonal chatter back and forth. And the T cell now clones itself. It becomes activated and it clones itself frantically. Very, very quickly, we end up with a large population of T cells. And the advantage of that is that these T cells now can go out and they can attempt to find a matching B cell. So here's the trick. Think back, and you'll remember I told you that B cells can actually ingest pathogen and display it. So they are also an antigen presenting cell. The B cell has encountered the same antigen, and it is displaying exactly the same portion as the dendritic cell displayed there. Now the B cell here, the T cell matched that on the dendritic cell, cloned itself, wandered around trying to match to B cells, and eventually found the B cell that was displaying the same matching antigen. As soon as that match occurs and this binding occurs between the T cell and the B cell, again, there's this same kind of chatter, this hormonal chatter that takes place between the T cell and the B cell now. And this activates the B cell, and it activates it really strongly. Immediately, the B cell begins to clone itself, to divide into thousands of B cells, all of them with this matching receptor. Okay, this B cell only has a receptor which matches exactly that little tiny portion of the antigen. Here it is binding to antigen there on an actual bacterium or whatever, but it's only binding to this very small specific part of the antigen, which we refer to as the epitope. So look what's happened. We had a dendritic cell that displayed that epitope. Only the matching T cell that matched to that epitope could bind. That cloned itself, thousands of them with receptors for that epitope, but they're only going to bind to a B cell that displays that epitope. And the only reason the B cell can display that epitope is because it had the matching receptor there. So this is the key to the specificity of the adaptive immune system. The response is specific to a very small part of the whole antigen. And only this line of B cells and T cells is activated that can match it exactly. But as soon as this matching takes place, as soon as this matching between the T helper cell and the B cell takes place, the B cell clones itself, and it clones into two populations. The first population are these very large cells that go into circulation and then end up in the bone marrow. And these are the plasma cells. And the plasma cells are going to produce antibody. And all that antibody is a soluble form of this receptor. There it is there. The B cells, the plasma cells now release masses of this protein. And what is its specificity? It carries exactly the same specificity because all it is a soluble form of this receptor. There it is there. And it's now, that antibody can now match that epitope on whatever it is, the pathogen, the virus, whatever, that initially initiated this whole process. So the specificity is carried on all the way through from what was displayed here, what activated the T cell, what activated the B cell, and now it's the same specificity there. So that's one population, the plasma cells. They eventually end up in the bone marrow producing masses of antibody. But the other population are equally important, and these are the memory B cells. And they are B cells which carry exactly the same receptor, but they are very long-lived. There's a lot we don't understand about memory cells, but we do know that they are very long-lived compared to all of the others. These plasma cells will last for weeks, maybe months, maybe a couple of months, but they don't last for decades. The memory B cells can last for decades, and what they're carrying a memory of is that. It's that they are identical in that their receptor is identical. So they are carrying a memory of this little epitope. And if ever the organism is exposed to that epitope again, these memory cells frantically start cloning themselves. We don't have to go through all of the permutations of trying this, trying that, trying this, trying that to match up, which takes 5 to 10 days. Instead, we'll get a reaction immediately. The adaptive immune system will jump into action immediately. It's re-exposed to the original epitope. Okay, so those are the helper T cells and how they interact with the B cells. But there's this other big group of T cells, which are the cytotoxic T cells, and they are also they're activated by dendritic cells. So they are going to react to the same epitope as the T cells and B cells. So we're going to watch as they are activated by the this is not a picture of the dendritic cell interacting with the T cell. That takes place in the lymph node. But the dendritic cell, when it exposes the little antigen fragment and excites the helper T cells, it can also interact with the cytotoxic T cells. And again, the cytotoxic T cells, they carry a specific receptor, each of them. And if the dendritic cell finds and encounters the correct matching cytotoxic T cell, the cytotoxic T cell is excited in the same way as the helper T cells were. It begins to frantically clone itself. But the cytotoxic T cells don't stay in the lymph node. They leave the lymph node and begin to circulate through the body, and they are going to interact with cells which are infected with that same pathogen. The way that they recognize the cells that are infected is as follows. Almost all of our cells can display can display protein from inside the cytoplasm. And we do it with this molecule. It's also an MHC molecule, but it is MHC class 1. And MHC class 1 continually displays just ordinary proteins, just whatever's going on in the cytoplasm. It will move small portions, especially of protein, to the surface and display it there. It's a way for cells to say, look, I'm self. I belong to you. The immune system is trained in ways we're not going to talk about, but the immune system is trained to recognize cells that belong to us. And this MHC class 1 is one of the ways in which our cells say, hey, it's fine. I'm you. I'm like you. Here are my proteins. My proteins are the same as your proteins. And there's no interaction. But if a cell becomes infected with something like a virus, that virus is going to cause all sorts of changes to take place in the metabolism of that infected cell. It's going to start producing unusual proteins that are not normal to the cell. And the MHC class 1 is going to start displaying those. It's not doing deliberately to advertise that it's been infected, but that is the end result. It actually starts to, the cell will now, it's displaying all its own proteins, but along with that, it'll start to display antigen derived from the infection, from the infecting particle. Usually, in this case, it's usually a virus. So now this MHC molecule displays antigen. And if this antigen that it's displaying is the same as the cytotoxic T cell receptor, then the cytotoxic T cell will bind to the cell and it will kill it. It kills in a particular way. It doesn't like cause the cell to just burst, die and burst and rot away. That would be chaotic, and you run the risk also of spreading virus, and also just releasing toxic byproducts of decay and everything else. Instead, the cytotoxic T cell causes the cell to actually essentially commit suicide. The cell begins to shut down all of its processes, all of its metabolic processes, wither away, and it dies in a programmed fashion. And this has a special name. It's called apoptosis. And apoptosis is actually a word which, a Greek word which refers to the falling of leaves in autumn. When the trees shed their leaves, that was called apoptosis. And it's a very good analogy, because the cells here that the cytotoxic T cell kills actually just wither away and then fall off, just are shed. And the cytotoxic T cell does this by releasing sets of very powerful enzymes called the perforins and the granzymes. I don't need you to remember those, but these cause pores to form in the walls of the cell, and it loses, the cell membrane loses its integrity, and the granzymes end up causing the cell to actually shut down and wither away, and it just is cleaned up, usually by macrophages and neutrophils that come in and clean them up. So the cytotoxic T cell has the same specificity. In our lymph node, the T and the B cells have the specificity for this one epitope, and they are now the B cells are producing antibody to that epitope, which is circulating in the body. At the same time, the cytotoxic T cells that have that same specificity are going out into the body, and they are seeking out those cells which display that particular epitope because they have been infected, and kill them. It's a very intricate, but it's a very cunning system. Okay, so if you have not done so already, I would suggest that you print out a copy of this diagram, which I drew up some time ago. This is the kind of summary diagram, which tells you this whole story. This whole interactive story takes place to produce the full-blown adaptive response. So we tell the story. We start the story with the entry of a new pathogen into the body. Let's say that this is a virus, because this system works both for viruses and for bacteria, but it works particularly well for viruses. A particularly strong response occurs when virus enters the body. Dendritic cells are unique in that they are able to actually take up virus, recognize that it's there, digest it, and display viral protein. Macrophages can't do that. They can do it with bacteria, but they can't do it with virus. So that's one of the reasons why dendritic cells are so very important. So we have a dendritic cell wandering around. It encounters virus in the body tissues, ingests the virus, and it digests it and displays it. And the dendritic cell immediately moves to the lymph node. It displays epitopes, small portions particularly proteins, derived from the virus, and it displays those using MHC class 1 molecules. So it displays them all over its cell membrane. When it enters the lymph node, T cells, both T helper cells and cytotoxic T cells, actually cluster around the dendritic cells. And what is happening is that they are attempting to match. They're attempting to bind to the dendritic cell display. But they can only do it if they themselves possess the exact matching receptor. Now, we produce T cells in the billions the whole time in our bone marrow. And when they are produced, their receptors are produced, the structure of their receptors is produced at random. So we've got T cells continual, this huge population of T cells, all with different specificities. A particular specificity is very rare. But using this system, by concentrating them in the lymph node and getting them to actually cluster around the dendritic cell, we're increasing the chances that the correct match will occur, but it takes time. That's why it takes like 5 to 10 days for this response to occur, because the adaptive immune system may even need to generate those T cells. They may not be there yet, but eventually a T cell with a matching receptor will be produced and will bind the dendritic cell. Immediately that binding takes place between the helper T cell and the dendritic cell. The helper T cell clones itself. It does it because the two of them exchange information. It's hormonal information. But it ends up activating this T cell, and the T cell now clones itself, and all of the clones possess that specific receptor. Okay, so we're going to leave that alone for the moment because we need to have a look at what is happening with the B cells. Also in the lymph node are these lymphocytes, the B cells. And just like the T cells, they also have receptors which can bind specific epitopes of the antigen. The first time a B cell encounters its specific epitope, the one that it can bind to, it will ingest whatever that is attached to. If it's a virus, it will ingest it. If it's bacterium, can also ingest it. And rather like the others, it will then digest it, cut it up into little pieces, and it will display the epitopes on its cell surface. Again, with MHC class 2. Now, this sounds like a rather strange thing to happen, but the importance of it is that this is how the B cell labels itself so the T cell can see it. Otherwise, the T cell can't see it, because the T cell is going to interact with that specific epitope displayed by the B cell. That's the only way a T cell can see things. That's how it saw the dendritic cell. It bound to it there through the MHC with the little epitope. That's how the T cell saw that was there and bound to the dendritic. Same thing here. It's going to the T cell with that specific receptor, matching the B cell receptor through the little display of antigen. That T cell now binds to its specific B cell. We say they are cognate. The T cell binds to its cognate B cell. And as soon as that happens, the B cell is powerfully kicked into cloning itself and starting to produce plasma cells, thousands of them. and the plasma cells produce antibody, and that antibody carries exactly the same specificity as was present on the B cell originally. So what is going to happen now? Well, that antibody is going to go into circulation and it's going to start reacting. But what does it react to? It reacts to exactly the same epitope. Exactly the same epitope as the T cell reacted to, exactly the same epitope as the B cell bound. Why? Because the B cell only has one specificity. Each B cell only has one specificity, and it will only produce this antibody with this specificity. That's how this kind of lock and key system exists, so that when we have virus or whatever, the antibody is very, very specific for certain molecular patterns on the virus or bacterium, and will only act there at the specific epitopes that it recognizes. So that's that story. This whole arm here, the release of antibody, this is called the humoral response. But there is another response as well, and this is the response that we've talked about with the cytotoxic T cells. And the cytotoxic T cells also interact with the dendritic cell. It's much the same story. A cytotoxic T cell has a particular receptor. If that receptor recognizes this epitope and binds to it, the cytotoxic T cell is activated, clones itself, leaves the lymph node and goes into circulation, and now goes out and seeks out cells which are infected, which are displaying exactly the same the same epitope. If it sees a cell that's doing that, it will kill it. And so we've got two things. This is called the cellular response. So in this adaptive response, we have these two powerful arms, antibody and then cytotoxic T cells, that are going to attempt to control the new infection, control the presence of the new pathogen. But at the same time as that is happening, each of these groups, the B cells, the cytotoxic T cells, and to a certain extent, not very much, but to a certain extent, the helper T cells, will also form memory. And they will form long-lived versions of themselves, so that if ever this is presented to them again, they don't have to go through this whole trying out for 5 to 10 days before you get the full response. Instead, the memory cells are sitting there ready. As soon as they see this, the whole system jumps into action. And within hours, not within days, but within hours, you're producing masses of antibody all over again, like that. Okay, so if you can understand this diagram, you've got the whole story basically clear for you. Are there questions? Okay. All right, so this obviously antibody is tremendously important. So we need to understand a couple of things about its functioning. Actually, its function is very, very complicated. The antibody is an incredible, it's not a molecule, actually there are many molecules that we regard as being antibody. But surprisingly enough, antibody is not like a toxin that will kill pathogens. It's not like it doesn't work like that. It works in many different ways, but one of the most important is that it will bind to pathogen. What is happening is the antibody, remember, is specific for a particular epitope. If it sees that epitope just on the outside of a pathogen, it will bind to it. And that binding has all sorts of effects. Let's just use this diagram. The very first effect, for example, with small things like a virus, or perhaps like a toxin, like an injected toxin, for example. Maybe a good example would be snake venom, for example, something like that. Antibody to that can bind to the surface. So here's the virus. Just remember, we've talked before about the functioning of viruses. All of that on the outside of the virus, there are a whole squad, many different proteins on the outside, which are functional. They're essential to the function of the virus, because these proteins on the outside, for example, are responsible for binding the virus to the correct cell that it can infect and reproduce in. Antibody can bind all of these proteins on the outside of the virus, and by doing so, they inactivate the virus. And so that's the first thing. This is called neutralization. The same thing will happen with a toxin. Antibody to a toxin can bind around the outside of the toxin, and it stops it from being active. It inactivates like that. The second thing to notice here, if you have a look here, you'll see here's an antibody, but the antibody has two identical binding sites, right? It's got one there and it's got one there. So let's take this antibody here, and we're going to use it like glue. It can actually bind another viral particle there. And that viral particle can have antibody attached to it, which can also bind other viral particles. Pretty soon, we're going to end up with a whole lump of this virus, antibody, all joined together. Virus is very difficult for the body to see. They're extremely small. Macrophages, for example, cannot reach out and engulf virus. But once we begin binding it all together like that, it becomes much easier for things like the macrophages to actually engulf and swallow it. This is a process which we call opsonization, and it's shown here as well for bacteria. And opsonization is the process whereby things can be labeled and made identifiable for ingestion by macrophages. And so you can imagine a lump of virus all tied together with antibody, or here a bacterium with which, let's put it this way, many pathogenic bacteria are able to almost camouflage themselves from the macrophages. They make themselves difficult to see. But when antibody binds them like this, they become extremely visible. And once they have this antibody bound like this, macrophages will actively chase them down and eat them. So it will, neutrophils, by the way. So this is the process of opsonization. Opsonization makes things much more visible and much easier to eat for cells like macrophages to eat them. But there's a whole other set of activities that the antibody can also generate. And these are really complicated. I'm only going to discuss them really simply. The first whole set of reactions, we need to cast our minds back to this. It's actually part of the innate immune system. These proteins which circulate in the blood, generated largely by the liver, called complement. And complement is an extremely powerful system, largely designed for dealing with bacteria. And it's part of the innate response, but it can be activated by antibody. So, for example, let's just say we've got a bacterial cell which is infected with an organism. The adaptive immune system has produced antibody to particular elements that are present in the cell membrane of that bacteria. And here's the antibody bound here. But the presence of that antibody actually can activate complement. So complement is in circulation, it is inactive. But when the complement can bind to antibody like this, it becomes activated. It does numerous things, the complement. But one of the really amazing things is that the complement proteins actually punch holes in the cell membrane of the bacterium. And when they punch those holes through, the integrity of the cell membrane is destroyed and the cell can no longer function. And so it dies. This is a simple example. There are many complex examples of interaction of antibody and complement. That's all you need to remember. This is one of the other ways that antibody activates the whole immune system, is by interacting with complement, which is part of the innate immune system. Okay, this diagram provides you with a summary, and in your text, this pertains to the development of memory. This is much the same story as in the diagram that I gave you earlier. All that I want you to take home, to emphasize here, is this portion of the response here, which is the generation of memory cells from the adaptive response. That whole complex adaptive response that we saw, each arm of it can produce memory cells. There are two memory populations that are of particular importance. And these are the memory B cells here, and then the memory cytotoxic T cells. There are some memory helper T cells produced, but in fact, the memory B cells can be activated without extensive interaction with the memory cells the second time around. So it's the memory B cells and memory cytotoxic T cells, which are really important, because if ever we are exposed to the antigen that settles this whole chain of events, the second exposure, there is an immediate massive population of active B cells and active cytotoxic T cells are produced. Okay, let me use this diagram to actually explain this. And this should also give you a good idea about how vaccination works. Because in vaccination, what we're doing is we're manipulating the immune system. We're giving the immune system a relatively harmless initial exposure to an antigen, and inducing the adaptive immune system and getting the immune system to produce memory cells. And those memory cells reside so that if we ever are exposed in real life to that antigen in the form of an infection, the immune system immediately comes into activity and it will eliminate the pathogen before we suffer any illness. So let's have a look here. This axis here gives us a concentration of antibodies circulating. We refer to that as the antibody titer. And that is if you have tests for COVID, one of the sets of tests you might have is a test for your antibody titer against the COVID virus. It would give an idea of how many antibodies you have circulating against the COVID virus. So that's the antibody concentration there. And this is just time along here in days. All right, so what we're going to watch, just look at the blue line first of all. And we're going to expose somebody to a particular antigen, antigen A. Well, initial exposure, if we remember, we're tracking antibody. Initial exposure, we don't see anything happening for quite a long time. It's 5 to 10 days on average for us to see the adaptive immune system produce its full-blown response. So after 5 days or so, we begin to see antibody beginning to build up. What is happening? We're developing a population of plasma cells and memory cells. And those plasma cells are increasing in number, and they're increasing in capacity to produce antibody. They produce more and more and more antibody that accumulates in the blood and in circulation. So we see an increase in the concentration. If after some time, this response, the adaptive response, is actually shut down, when it is no longer needed, there are specific mechanisms to silence the adaptive response. It's not something you want to go on and on and on after it's needed. It takes a lot of energy. It's a complex process, and it has also side effects as well. Part of the side effects are things like increased temperature, et cetera, et cetera. So you don't necessarily want this whole response to persist longer than it is necessary. And there are ways the immune system actually has of recognizing that it is no longer necessary and shutting the adaptive response down and leaving only the memory cells to be active. So what we observe is a decrease in the antibody levels. As the infection is cleared and everything returns to normal, we don't carry on producing masses of antibody. The antibody levels decrease. Here, however, is a second exposure. This is something which occurs in us very frequently. For example, every winter, there are cold viruses which circulate, and most of us have a cold once or twice a year. When you have a cold induced by a particular virus, you know, you have 5 days of feeling really not so good. And then some days when you know you're recovering, you've still got some symptoms, but you're actually recovering, and then everything's gone. This is what's happening. Your body is producing antibody to that cold virus. You're reacting to it. The virus is eliminated around about here. And once the virus is gone, your immune system says, okay, now we're going to quieten everything down. And the antibody levels to that virus fall. They're no longer needed. But you have developed memory. You've developed memory of that cold virus. You can bet in one winter, you're going to be exposed to that cold virus again. And when you are exposed this time, you don't have that long wait. Instead, within days, in fact, within hours, then within days, you produce a massive antibody response. Look at this. Here is the original. And that was perfectly effective, right? Here's your secondary. This is called the secondary response. Look at it. It's huge. And I'll tell you something else we have not discussed is that antibody can be improved. As this process goes on longer, the ability of antibody to work is improved, and you produce more and more kinds of antibody that can have let me explain this way. You start producing antibody, for example, which can be excreted in your saliva, in your mucus, across the gut wall, into breast milk. You start to produce, as this goes on, you produce these antibodies, all with the same specificity, which can move into different parts of the body. And all of that takes time. But now we leap into the full-blown response and produce all of our really good antibody, all with really elaborate functions immediately. Again, it eliminates the pathogen, and once it does so, the antibody levels begin to decrease again. They decrease more slowly the second time, but they decrease. If you have a third exposure, you get an even bigger response. So this is a very powerful, very, very sophisticated system, and it works incredibly well. But it is highly specific. It's very, very specific to that particular antigen, and usually to that one particular pathogen. It does not give you blanket protection. It gives you protection for things which may look very, very similar, but it doesn't give you blanket. Because you've reacted like this to one virus, you have no protection against another one. Have a look here. Here we're going to introduce a second antigen of some sort. This is antigen B. We've got this wonderful response to antigen A. But now let's introduce antigen B that looks different. What happens? Look there. What is that? Well, it's exactly the same as this response here. You have exactly the same. This is a new antigen. You have to do the whole thing all over again. Okay, now you've got to regenerate all of that, all of those processes. But again, you're going to form memory now for antigen B. And if we have a second exposure to antigen B, the response would look much like this. So this is the principle of vaccination. What we do in vaccination is we induce this primary response, and we do it in such a way that there is minimal impact on the functioning of the body. We usually do this by exposing, let's take COVID as an example. In all of the COVID vaccines, in one way or another, what they are doing is exposing the body to what the proteins look like on the outside of the COVID virus. So this induces a response just to that particular protein. People in general, they don't feel very much, especially this first injection. Let's say we're doing one of the ones where there's 2 injections. This is the reason. We do injection 1. Injection 1 would be here. It induces a response. The body produces antibody, learns what that antigen looks like, and forms memory. As most of the vaccines now, you have to go back for your second dose, come back for your second dose. Here's the second injection, and boom, your body recognizes and produces a massive response, a massive improved and improving response to the presence of the antigen. This is one of the reasons, by the way, why some people, I didn't, but some people actually, with their second injection for the COVID vaccine, they actually feel ill. The reason is simply that the immune system is kicked into action. It's producing masses of antibody, but sometimes it produces other physiological effects as well. They're temporary, harmless, temporary, might be a little bit of an inconvenience, but you're left with excellent memory. So that if you ever in real life encounter the virus, you will produce a response like this immediately. You don't have to go through this time, all this time doing it. You'll do it straight away. Okay, so there are different, that is actually what we refer to as immunity. We say we have immunity once we have acquired that memory, and if ever we're exposed, we eliminate the threat immediately. There are different kinds of immunity. The first is we call passive immunity. And this is immunity which is conferred from mother to an infant. It takes about 6 months after birth for the immune system to reach its full maturity. So infants are at some risk of infection, some greater risk of infection during those 6 months. But they are covered by the fact that any antibodies which are circulating in the mother's bloodstream can percolate across the placenta and accumulate in the infant. So the infant is born with a set of antibody, which is acquired from the mother. And that is referred to as passive immunity. It fades after about 6 months, but by that time, the infant's own immune system is sufficiently active that it can counter infections. This passive immunity is also interesting in the current COVID pandemic, because there have been some attempts to take blood from people who have recovered from COVID and therefore are assumed to have high antibody levels in their blood, and to actually extract those antibodies and inject them into people who are in the earlier stages or severely ill from COVID. It appears to be a somewhat hit and miss operation. In some people, it appears that it does indeed have good results. In others, it does not. And that probably depends on the antibody that the donor produced. Maybe sometimes that antibody, it doesn't work that well in somebody else. Nonetheless, this is a therapeutic method, is extracting antibody from somebody who has recovered from a disease and trying to inject it into somebody suffering from the disease. And this is another form of passive immunity. Vaccination, or immunization, is called an active immune response. This is where we actually train the memory cells. The active immunity, it naturally occurs because of the presence of memory cells. We can manipulate that artificially in the process of vaccination. Now, the question is, let's just go back here, is what do you immunize with? What do you use to do this vaccination? Very often, we'll actually use a harmless form of the pathogen that we want to immunize against. That has been a standard way of vaccinating for many years, more than 100 years. Sometimes they will take a virus and they will weaken the virus somehow so that it can't cause an infection, but the body can learn to recognize it. The other way is to use an organism which is very, very similar, but harmless. That is the classic way, for example, that smallpox was vaccinated for. We used another virus, a cowpox virus. Looks to the body, looks just like smallpox, but it does not produce disease in humans. But it trained the body to recognize the smallpox virus. In the case of COVID, what we're doing in one way or another, all of the vaccines try to get the body to recognize those outside proteins on the virus called the spike proteins. Those spike proteins, their role in the life cycle of the virus is that they are how the virus binds to cells to infect them. So if we can produce antibody to those proteins, we can stop the virus from being able to infect cells. Okay, I know I'm going to go a little bit over, but let me finish this. I'm only going to discuss our last topic, which is allergies, very, very briefly. I'm more because I know that some of you who may have allergies may be interested. And in discussing allergies, we have to go all the way back to the beginning when we talked about innate immunity, to that set of cells called the mast cells. Now, mast cells are resident all over our bodies, in all of our tissues. They contain histamine, and they respond to certain specific signals by releasing the histamine. Now, an allergy is produced by a substance called an allergen. And an allergen is something which is essentially harmless, but which produces a response as if it was an antigen. And the response of the body to that allergen is as if it was something noxious, as if it was a pathogen or something like that. So we've got something completely harmless producing a reaction as if it was harmful. Just think of pollen. What can pollen actually do to you? Pollen can't infect you. It can't hurt you. It can't do any of those things to you. And yet, in many people, when it lands in their eyes or when they breathe it up their nose, it produces an intense reaction of hay fever, of swollen eyes, weeping, runny nose, blocked nose, etc., etc., etc. Why? Why would pollen do that? Well, the answer lies in the activity of the mast cells, because mast cells have a very peculiar property, and that is that they pick up antibody. They pick up many, many, many different antibodies, antibodies with many, many different specificities to all sorts of different antigen, okay? And they take this antibody. So this antibody is being produced in response, the adaptive response. Something has come in and has induced the production of antibody, and the mast cells will take some antibody and plug it into its cell membrane and use it as a receptor. And if ever it sees this allergen, this antigen again, during an infection, for example, during a secondary infection, the mast cell will, when it binds an epitope there, it will release its histamine. Well, that's very useful. That's very useful. That'd be a very useful adjunct to our adaptive and memory responses, right? This is kind of like mast cells have memory of what antibody has been produced. Here's the problem, is at some point, like you got pollen in your eye, and your adaptive immune system, for some reason, thought that this was pathogen and produced antibody to it. You probably didn't, the first time it happened, you didn't even notice. This is a quiet little thing that happens in your immune system, in your reactive immune system, produces antibody to pollen, but that antibody now plugs itself into the mast cells as a receptor. And the second time you see the pollen, all your mast cells, like in your eye, they see that pollen, they bind it as antigen, and they expel all their histamine and other substances as well. So what does histamine do? It dilates the blood vessels. Your eyes get bloodshot. The blood vessels become leaky because of the histamine. They start leaking serum. Your eyes swell. In your nose, the same thing happens. Your blood vessels expand. Your nose gets blocked. Your nose starts running, etc. All of these are effects of the mast cells releasing histamine. So this is a huge nuisance, right? That's a nuisance, like for in your eyes. Those kinds of reactions are localized, but they're not life-threatening. Here's another problem, and that is that if instead of just having a localized exposure, you get a global exposure. For example, let's say that the allergen, the original, was something like a bee venom. Okay, so how's bee venom work? It gets injected into you. It circulates throughout your body. Mast cells all over your body can develop a sensitivity to bee venom. The second time you're exposed, if you've done this, then all of the mast cells all over your body release their histamine all at once. And as a result, all the blood vessels in your body dilate. Your blood pressure crashes catastrophically. All the blood vessels begin to leak. In the lungs especially, they begin to leak fluid. The lungs begin to fill up with fluid. The throat begins to swell up. All of these, you're now in a life-threatening situation. And that is the process of anaphylaxis, anaphylactic shock, which is a life-threatening situation that arises because of circulation throughout the body of a toxin, of an allergen, such as bee venom or something like that. The same thing also can occur with some things which are consumed. Peanut allergens, for example, diffuse across the gut and go into circulation and can do the same thing. So that, I'm afraid, is all that I'm going to be able to say about allergens. Okay, and we'll leave it there for today. Are there any questions before we go? If not, I say goodbye to you. Yeah.
Have a good weekend, Professor.
You
man,
too.
I see you on Wednesday, right?
Yes, yes.